Welcome to the Microbiology Laboratory. In food, water, and pharmaceutical microbiology and clinical laboratories, it is important to determine the number of microorganisms. This process is known as enumeration of microorganisms. Enumeration helps us determine microbial quality, contamination level, safety of food and water, effectiveness of sterilization. In this video, we will learn three important methods of microbial enumeration: plate count method, membrane filtration method, most probable number method. Enumeration means estimating the number, counting of microorganisms present in a sample. Enumeration generally measures viable microorganisms capable of growth. Depending on the method used, microorganisms may be counted as colony-forming units (CFUs), most probable number (MPN), cells per milliliter. We use standard plate counts for food and milk. Membrane filtration is ideal for beverages, while MPN is best for water. The standard plate count method is based on a simple principle. Every viable microorganism, when provided with suitable nutrients and incubation conditions, grows into one visible colony. Each colony develops from one microbial cell or a group of cells and is known as a colony-forming colonies. We can estimate the number of viable microorganisms in the original sample. Welcome to our lab session. We have all our materials ready. Here are the agar plates and spreader. Let's begin our sterile dilution. Take 10 grams of food sample, add it to 90 milliliters sterile diluent. This forms the first 10-fold dilution or 10⁻⁻1 dilution. Transfer 1 milliliter from the first dilution into the next tube containing 9 milliliters of diluent. Mix thoroughly. Repeat the process until the required dilution is achieved. Each transfer reduces the microbial concentration by 10 times. In the spread plate technique, transfer 0.1 milliliter of diluted sample onto a solidified agar plate. Spread evenly using a sterile L-shaped glass spreader. Incubate the plates upside down at 35 to 37 degrees Celsius for 24 to 48 hours in BOD incubator. After incubation, colonies become visible. Only plates containing between 30 and 300 colonies are considered suitable for counting. Too few colonies decrease accuracy. Too many colonies overlap and cannot be counted reliably. 300 reliably. Let's calculate CFU. Here is the formula. For example, with these values. That gives us this final result. Here are the advantages of SPC. It counts only viable organisms, it is simple and accurate, and it is widely accepted. The membrane filtration method is based on physical retention of microorganisms. A sterile membrane filter with pore size 0.45 traps bacteria while allowing water to pass through. After filtration, the membrane is then placed on nutrient medium. Trapped bacteria grow into colonies. Let's demonstrate membrane filtration. Sterilize filtration apparatus. Place sterile membrane filter. Filter measured sample volume, usually 100 milliliters. Wash funnel if required. Remove membrane using sterile forceps. Place membrane onto selective agar. Incubate. Count colonies. Let's calculate the CFU. Use this formula to find it. For example, 25 colonies gives 50 CFU. This method is highly suitable for water samples containing low numbers of microorganisms. This method offers key advantages. It is sensitive and rapid and is accurate. However, there are limitations. It's not for turbid samples. Membrane clogging can occur. The most probable number method is a statistical technique. Instead of directly counting colonies, estimates microbial population based on the number of positive tubes showing growth after inoculation. Prepare serial dilutions. Inoculate tubes with different sample volumes 24-48 hours. Observe acid and gas formation. Positive tubes indicate microbial growth. The combination of positive tubes is recorded. This combination is compared with the standard MPN statistical table. MPN is especially useful when microorganisms are present in low numbers or cannot be counted reliably on agar plates. Let's discuss some advantages. It is suitable for low bacterial counts. Consider the limitations. It can be time-consuming and it requires many tubes. Microbial enumeration assesses quality and safety. Standard plate count, colony counts, membrane filtrate samples, most probable number offers statistical estimates. Thank you so much for watching.